Welcome to Science News Review. For July 20, 2026, we are looking at how science is reading the code of life in unexpected places, from the complex genetic markers of mental health to the microscopic traces of ancient humans left on cave walls. We begin with a major step in understanding the biological basis of psychiatric conditions. For a long time, the conversation around borderline personality disorder, or BPD, has focused heavily on psychological and environmental factors. However, a massive genome-wide study published in Nature Genetics is now providing some of the first significant evidence of a genetic component. By combining data from 27 different studies, researchers analyzed more than 6 million genetic markers across a vast group of people, including over 1 million individuals who do not have the condition. The results identified 11 specific locations in the human genome associated with a risk for BPD, including 9 functional genes. While 6 of these sites held up during replication analysis, researchers noted that 5 other identified locations still require independent verification to confirm their role. There is a notable gap between these findings and previous estimates. These specific genetic variants account for about 17% of the inherited risk of BPD. That is significantly lower than the estimates from earlier family and twin studies, which suggested the heritability could be as high as 69%. This discrepancy suggests that while genetics play a role, the picture is far more complex than a single set of markers might suggest. It is also important to note the limitations of the current data. Because the study participants were primarily of European ancestry, these findings may not apply to other populations. Furthermore, because enrolling people with BPD in large-scale genomic studies has historically been difficult, the researchers suggest that while this approach shows promise, similar to how we study schizophrenia or depression, there is still much more work to be done to understand the full biological landscape of the disorder. Moving from the complexities of mental health to the possibilities of reproductive medicine, researchers have reported a significant milestone in the treatment of male infertility. At a recent meeting of the European Society of Human Reproduction and Embryology, scientists presented findings from a case involving a man who has regained signs of sperm production through a testicular tissue transplant. The patient's journey began when he was 10 years old. Before undergoing chemotherapy for sickle cell disease, doctors had removed and frozen samples of his testicular tissue. These samples were recently grafted back into the man's scrotum and his remaining testicle. The tissue was allowed to incubate for a year, allowing it to be exposed to the body's adult hormones. The results, released as an unreviewed preprint, are encouraging. Researchers identified sperm-producing stem cells and signs of active sperm production in several of the transplants, including the observation of a single mature sperm. While this is a major proof of concept, the clinical application is still in its early stages. For now, researchers have paused further analysis of the patient to preserve the remaining tissue grafts. The hope is that they can collect enough sperm to eventually attempt in vitro fertilization, potentially allowing men who lost fertility due to early life medical treatments to have biological children. Finally, we look at how the very surfaces of our history might hold the secrets of our ancestors. In a project known as First Art, archaeologists have successfully recovered ancient human DNA from cave walls in Spain and Portugal. By collecting over 100 samples from 11 different caves, the team identified 5 samples that contained human DNA. One of the most intriguing aspects of this discovery is where that DNA came from. While some samples were found on unpigmented rock, and others appear to have been carried into the caves by water or wildlife, researchers found a sample at the Escurial Cave in Portugal that lacked animal DNA. This suggests the traces may have come from direct human contact, such as touch or breath. It is important to manage expectations regarding the connection to the cave art itself. Currently, the link between the DNA and the paintings remains unproven. Only one of the positive samples was recovered from a painted surface, and researchers caution that the DNA could belong to anyone who passed through the cave long after the art was created. However, the implications for archaeology are profound. If this method can be refined, it could allow scientists to study prehistoric populations, including the roles of women or Neanderthals, by reading the genetic traces they left behind on the very walls they once stood before. Whether it is the potential for medical breakthroughs in fertility or new ways to map human history, these stories remind us that the answers to our biggest questions are often written in the smallest details of our biology.